is we've all known, I think, at least in this group, of the importance of vitamin C in the body and of the fact that most people are genetically deficient, not 100%, in the ability to make vitamin C. It should be very exciting to realize, and I'll show you the evidence, that there are ways to overcome this genetic shortcoming and to allow people to start making vitamin C most of the time. Like all my other presentations, I have many different references, and all you see is a number. You take the number, go to this website, put the number in, and you'll go straight to the abstract and sometimes the entire article discussing the fact that I'm citing. Most of this information initially came from the research and brilliant mind of my colleague, Mr. Bill Sardi, who unfortunately passed away several months back, but most of this information is due to his diligent research. Even today, it's pretty much assumed by people that are aware, scientists that are aware that regular animals make vitamin C in their liver, that human beings cannot make it because they have a defect in the actual sequence of the nucleotides in the DNA. In most of the wild animals, they not only make vitamin C on a regular basis around the clock, along with cortisol released from the liver, they make a lot of vitamin C and release that into the blood in the face of acute infection and acute oxidative stress. I'm going to show you some of the evidence in the scientific literature that indicates the inability of human beings to make vitamin C is not because the sequence of nucleotides in the DNA is missing or in error, but the interpretation, the transcription of those DNA nucleotides is what's the problem. Because I'll talk about this a little bit more, but you have genetic defects and you have epigenetic defects. Genetic defects means you no longer have the proper sequence of nucleotides coding for a protein in the DNA. And epigenetic means you've somehow lost the ability to transcribe correctly and accurately the RNA that comes from that DNA to make a protein. And as we'll see as we go on, these epigenetic defects, this acquired inability to properly produce a protein from a correct DNA strip, is actually much more common than we think and is almost always acquired after birth. Like we've talked about in other presentations, all damage in the body is called by oxidation, and all good things in the body are pretty much caused by repair of that oxidation. And so it appears that agents that can target these sequences that need to be properly read to make a protein end up being oxidized, and we have found agents that are able to repair this. To be specific, the protein that's directly involved in converting glucose into vitamin C is called L-gulonolactone oxidase, abbreviated as GULO, G-U-L-O. And this is the final enzyme in a sequence of 4 enzymes that are needed to make this conversion. All human beings, the first 3 enzymes are in good shape. It's just this 4th enzyme that we have problems with. Now, when I talk about a DNA sequence being read, producing messenger RNA, and then that messenger RNA has a ribosome that rides along it, producing the different amino acids that make the final protein. It might be a little amusing, but you're literally having a ribosome ride along the messenger RNA like a train on a track, producing the protein, and the train encounters a big lump in the track or like a piece of gum on a zipper and stops. When you can get it to jump over that gum and complete the transcription, you can still make the protein, which is what we'll be talking about. When you're able to bypass or go over this piece of gum on the zipper, this is referred to in the literature as stop codon read-through. Now we know of some other inherited diseases that pretty much medicine thinks are fixed problems with DNA, are also epigenetic problems with abnormally placed stop codons, and this includes beta thalassemia and cystic fibrosis. These studies also show that there are drugs, in this case aminoglycosides, that can jump over the piece of gum and restore protein synthesis in the RNA strands and help lessen and sometimes eliminate much of the problems of these inherited diseases. It's very important to find agents that have little or no toxicity at all to stop this epigenetic defect, because drugs like aminoglycosides have significant toxicity, and you don't want to take those for extended periods of time or even for life. In fact, now, and you see these references that you can go to, there are quite a number of natural and non-toxic molecules that have been discovered which appear to allow jumping over the phenomenon known as stop codon read-through. I'll read this directly because it's very important, and that is the concept of acquired epigenetic defects is further supported by infants who are not born with thalassemia, but later develop thalassemia along with the inability to synthesize enough hemoglobin to survive without need for repeated blood transfusions. In the test tube, resveratrol, a polyphenol in grapes and wine, has actually been found to induce the production of fetal hemoglobin in patients with thalassemia. Of course, even though it's not 100%, it was shown that fully 50% of these thalassemia patients completely no longer needed repeat transfusion after resveratrol treatment, indicating the epigenetic defect had been overcome. Guinea pigs are sort of the animal equivalent of human beings, and they can't make vitamin C as well, but they do have the proper genetic code to make it. They just also don't have the ability to transcribe it into the protein. However, if you look at these references that I have here, there is a small minority, but nevertheless, animals and people exist among these guinea pigs and people that do actually synthesize vitamin C in their livers. It would appear that babies regularly make vitamin C, which would mean it's not a fixed genetic defect, but an epigenetic defect. In the womb, the baby makes large amounts of vitamin C and ends up with brain levels of vitamin C 400 to 1,000% higher than most adults. Similarly, umbilical blood appears to have most of the time fourfold, 400% more vitamin C than the maternal blood. Information on babies indicates that breastfed babies, even though all their nutrition is coming from mama, they maintain blood levels 200% higher of vitamin C than of the mother, and it was not reflective of the vitamin C levels in the breast milk. Very difficult to imagine how these levels of vitamin C inside the fetus and inside the babies after they're born could be coming from anything other than new synthesis of vitamin C in those very young livers. Children, well beyond infancy. In this particular tribe, Bantu tribe children in South Africa, even though they were taking in an incredibly tiny amount of vitamin C daily, 3 to 8 milligrams per day, the symptoms of scurvy never appeared and were never noted. In writing the paper, the investigators themselves concluded that the only logical conclusion is to postulate that there is an endogenous or internal production of vitamin C in these children. Let me say, and this is my opinion, I think these Bantu tribe children were effective at making vitamin C because they lived far away from the enormous amount of toxins that we see with children in much more modern civilizations in both food, air, and water. And yes, some adults make vitamin C. They have, in the past, done studies on adults by seeing how long it takes for them to stop spilling vitamin C in the urine and to start developing symptoms of scurvy and vitamin C depletion. One study, an adult female, actually continued to show high blood levels even as the vitamin C intake got progressively lower and lower. Another woman went 149 days, almost half a year, without any dietary vitamin C, and she developed no deficiency symptoms at all of vitamin C. Similar findings to these isolated adults have been seen also in isolated guinea pigs, some of which appear to make normal levels of vitamin C. To re-emphasize this primary point of genetics versus epigenetics, epigenetics refers to the variable genetic expression due to modifiable factors that can block or impair the accurate transcription of messenger RNA coming from the DNA. And to boil it down to 4 words, and it's always good to be short and concise, epigenetics means correct code, incorrect translation. So reflecting on all the studies that we've seen on fetus, babies, children, and adults, it appears certainly that nearly everybody initially has the ability to make vitamin C at a very young age and eventually loses it, presumably due to their unique but unavoidable exposure to the wide range of toxins that we're exposed to on a daily basis around the world. Now, in a pretty incredible study done in 2017, the polyphenol hydroxytyrosol, which comes from olive leaf extract, was found to double the average levels of vitamin C in healthy volunteer subjects. This doubling of concentration persisted because 45 milligrams a day of this polyphenol showed that at 1 month, the levels were doubled, and at 2 months, another test was run and they were still doubled. As a result, and it shouldn't come as a surprise, the body-wide effects of hydroxytyrosol administration appear to closely parallel the effects of vitamin C administration. Now at the Reardon Clinic, Dr. Ron Honeyhackey ran some additional tests on oral hydroxytyrosol. 5 volunteers, they were given 50 milligrams of hydroxytyrosol a day. And after only a week, depending on the person, the blood level of vitamin C was up between 50% and 200%. Not surprising, the lowest levels of vitamin C showed the most profound response in one smoker who had quite a depressed vitamin C level at the start, got a dramatic increase in the vitamin C level on the hydroxytyrosol. In 4 of 5 volunteers, a clear-cut elevation of blood vitamin C was seen the day after the first dose. This is important because even though my recommendation to anybody who wants my recommendations is that you should be on hydroxytyrosol, basically a quality olive leaf extract product for life, if you don't have the inclination to do that, or it's too expensive for you, you should still have a bottle around because it appears that you can cause a quick response of vitamin C synthesis very quickly, and this can help you get over acute infectious syndromes. It also appears that the vitamin C blood levels stayed elevated in this particular case for a full 10 days after the 2-week administration of the hydroxytyrosol. Though we don't have the studies to show it, I seriously doubt that taking hydroxytyrosol and then stopping it is going to give you a lifelong result of increased vitamin C synthesis, because whatever caused the epigenetic defect, whatever toxins that you faced are going to continue. So it's my impression, without further studies, that the hydroxytyrosol in the form of olive leaf extract should be taken for life for this incredible benefit of starting vitamin C synthesis again. 2 people, namely Dr. Honeyhackey and myself, who had been taking hydroxytyrosol for some time, we decided to see whether a large dose of toxin, which is going to use up vitamin C and depress the levels, what that would do inside us, assuming that our livers were able to respond to that toxin and start making more vitamin C rather than less. We were all set up to get vitamin C blood levels drawn. After a large amount of alcohol was ingested, blood levels were taken, and they initially showed a severe depression, which meant the alcohol was metabolizing the vitamin C, but it was rapidly followed by a large rise in vitamin C that stayed high for nearly 18 hours after the alcohol ingestion. Remember that vitamin C is not stored in the body. And if your liver cannot make vitamin C, then when you're exposed to a toxin, all it does is drop the vitamin C until you take more vitamin C orally. I'll show you on the graph on the next slide that the vitamin C level rise was quite significant, and it persisted for 18 hours. And the next morning, both Dr. Honeyhackey and I felt not just good, but we felt particularly good, which would be consistent with having even more vitamin C in our body than normal. These two, and on this slide, alcohol is in blue, alcohol blood level is in blue, vitamin C blood level is in red. Just like we noted, the immediate effect in both Dr. Honeyhackey and myself was a quick and massive drop of vitamin C as the alcohol reached peak levels. Just the opposite occurred with a little more time as the liver realized, if you will, that the toxin was present and then started making more vitamin C, which caused the red line to go up while the blue line went down. Now, the short and long-term health benefits of this cannot be overemphasized. There is no medical condition or disease, no matter how well it's being treated, that will not be even better treated when the liver is making vitamin C on demand with increased amounts when there's more oxidation and more infection present in the body. Many different animals can make their vitamin C. A goat, about the size of an average human being, can make 13 grams, 13,000 milligrams of vitamin C a day. If your liver is making 13,000 milligrams of vitamin C per day and releasing it directly into your bloodstream, this is not remotely approached by taking 13,000 milligrams of vitamin C orally because of absorption problems and many other problems. You would have to take a much, much, much larger dose of vitamin C orally to even come close to what you can achieve when it's being directly released into the blood. I want to address the incredible synergism between the two most powerful and naturally occurring anti-inflammatory agents in your body, vitamin C and cortisol. If your liver is working correctly, like it is in the wild animals or in a human who has overcome the epigenetic defect, two things happen when you have a large amount of infection or oxidative stress in the blood. Presence of large amounts of oxidative stress in the blood causes the simultaneous release of large amounts of cortisol from the adrenal glands, while the liver is releasing large amounts of vitamin C into the blood. The liver is making vitamin C from glucose. So this is allowing your body to use up glucose stores properly rather than let them stay elevated and cause long-term diabetic and other sugar storage problems. This is especially important, and I don't have time to go into it in detail, but it would appear that a primary role of cortisol, maybe the primary role of cortisol, is to force the uptake of vitamin C into the cells of the body. This means that anything that can cause the human liver to start making its own vitamin C results in a significant increase in intracellular vitamin C levels throughout the body because the cortisol is being released from the adrenal glands at the same time. So to give it a little bit of a sequence here, this is what should happen when your liver is making vitamin C. New blood toxins result in both vitamin C and cortisol release, which immediately causes a massive increase in the cellular uptake of vitamin C throughout the body. Interestingly enough, the cortisol also provokes new glucose production, which allows it to further feed and sustain the ability of the liver to make more vitamin C. Just like it worked in Dr. Honey Hackey and myself, this increase in vitamin C synthesis and it's being pushed into the cell by the cortisol continues until the oxidative insult has been completely neutralized. In our case, the vitamin C level stayed up for 18 hours and even had us feeling better than normal before it came back down again. Very important points here. And I have a lot of information on this, but it's beyond the time that we have here. Infections of all variety occur because cellular levels of vitamin C have dropped too low. Patients that have little or no adrenal function, meaning they can't make cortisol, the primary concern is life-threatening infection because there's nothing available to push the vitamin C into the cells. It's well established that old patients develop sex hormone deficiencies, they develop thyroid hormone deficiencies, and guess what? Most of them develop a mild to moderate to severe degree of cortisol deficiency as well. It's not normal to get a cold or the flu, or even COVID for that matter, and keep it for more than 24 hours if you're making enough cortisol and if you're making enough vitamin C. If you keep a cold or flu or COVID for days to weeks, you are cortisol and vitamin C deficient. And here are 2 articles I've written on this that go into a lot more detail on this interaction and synergy between vitamin C and cortisol. With regard to hydroxytyrosol, you generally want to get yourself in a dose level of 45 to 50 milligrams daily. The best known source of hydroxytyrosol is olive leaf, and to a much lesser extent, olive oil. Hydroxytyrosol, in addition to the effect it appears to have on allowing the synthesis of vitamin C in the liver, it's also by itself also an extremely powerful antioxidant. Olive leaf, and I plan on doing much more research on this, also has an incredibly large number of highly beneficial substances in it, including 2 substances known as oleuropein and tyrosol. At this point in time, my advice is to take an olive leaf extract supplement that can give you 45 to 50 milligrams of hydroxytyrosol a day or more, but to take the whole extract. It's incredible, and there's a very large amount of literature talking about the benefits of olive leaf extract for just about every condition under the sun. Some sensitive stomachs require that it be taken with a meal, but most people are not that sensitive. I've long talked about what I consider the 4 most important supplements, which are magnesium, vitamin D2, vitamin C, and vitamin K2. I would wholeheartedly add to that list olive leaf extract as being something you should never go without taking if you have availability and if it's not too expensive for you, because its health benefits, in addition to what it does to vitamin C synthesis, are truly extraordinary. And that's it.